The state of the art. Good evening, everyone. I am replacing Professor Di Francia, who is not here due to health problems. I'm Giancarlo Blasio. I'm specializing in medical genetics at the two universities of Naples, Federico II and Luigi Vanvitelli universities in Campania region. Today I'm going to talk about a presentation on polymorphisms and the role that this might entail in the future as regards the vaccine practice. So with this presentation, actually, this could be food for thought to better understand how genetics can be inserted in vaccinology research. I'd like to resume a slide that Professor Del Buono has already shown regarding personalized medicine. We live in a personalized medicine context.
Evidenziate con la genetica.
And thanks to the discoveries of molecular biology, there are some differences, especially according to the genotype of the persons. So this entailed that the higher knowledge of DNA and the differences and the genetic features, personal features, allowed to emphasize how different persons with a different genetic background do not comply in the same way with therapy. So this is why we need personalized medicine. Of course, this could not be possible without the advances in technologies, these breakthroughs that allowed us to better understand more into detail and into depth the DNA. So we talk about arrays technique. The new shift within next generation sequencing, the sequences of a new generation that allowed and allows today to sequence genomes at very low cost in a very short time. As for vaccines, probably this input towards personalized medicine has been delayed. This is due to the fact that the strategy of vaccine that has been adopted to date is, by the way, efficient. So most of the vaccinated population has a total immunology in this kind of point of view. But there are certain cases in which this does not happen. So we are at two extremes. On the one hand, persons who do not have a total immunization. On the other hand, the so-called adverse reactions. So due to the scope and the application and the use of vaccines, if we were to compare with other drugs, it's a matter of, analyzing the details. This could be represented with a Gaussian curve. This phenomena could be at the very extremes of this curve, but it is, important to understand why we have this trend. And so we can try to improve continuously. There are different reasons. There's not a single answer to this problem because vaccination, the vaccine response is very complex. It depends on the balance of many different factors of the vaccine and the host. In the background of personalized medicine, we can, get to know and control better and better the genetic part, the genetic background. And so today, we can base ourselves on the knowledge of personalized genetic characteristics. Today we have a whole set of tools, a whole set of weapons, if you want, that allow us to understand and to know a person at different steps, at different levels, from the most intimate part, that is the genomics and DNA, to the overall global picture of metabolomics. So we cannot neglect the role of the ITC technologies. Bioinformatics is increasingly important. The data banks are very important and detailed. They provide us lots of insights. So all this knowledge today put together are very important because they allow us to start multidisciplinary studies that to move on with my slides if possible. Now it works. So this allows us to, according to the genetic individual characteristics, to develop new vaccines. So, thanks to these studies, investigating the DNA of the subjects involved, we can discover new targets, new biomarkers, and then we got the possibility to develop new vaccines. So this whole framework, all these new studies could allow to move from the empirical vaccinology, so from the strategy of one size fits all kind, I give the same dose to everyone. In theory, these multidisciplinary studies, together with the knowledge of the genetic individual peculiarities, can allow us to move towards personalized therapy, also within the scope of vaccines. So different persons, different vaccines in terms of dosage and the vaccine itself. So, but before that, we of course need to understand the genetic differences of persons, and these are mainly made up of DNA polymorphisms. It is estimated that there's about one polymorphism every 1,000 DNA bases. This means that if you calculate the whole number of bases of the genome, this means that each individual has a unique genetic pattern. So polymorphisms can be single sequences like SNPs, FLP, or tandem polymorphism. This is a pair or a triplet of nucleotides that repeats more than once in our genome. So each person has a different number. Today, polymorphism play a role and have an impact from the clinical viewpoint of a different kind. We move with the paternity tests that are used today. This is a table that shows about 50 genes whose polymorphisms are associated to the development of cardiovascular diseases. Again, we have one of the most known HLA locus immunity and vaccines for which some alleles have been demonstrated to be related to the development of certain pathologies. And so specific tests have been developed for specific pathologies. And then, of course, in this table, I've shown also the MHFR gene, which is one of the most widely known genes from this point of view that is involved in the homocysteine metabolism. So the homozygous or heterozygous polymorphisms, these are two kinds of polymorphisms, and therefore there will be a therapy with folates. So to date, for different scopes, they play an important role in the clinical setting. So the next step, what happens for vaccines? Today, for vaccines, we do not have anything concrete, but many studies are underway creating associations between single nucleotide polymorphism and the vaccine response, which has different kinds of responses according to the vaccine. So what kind of studies are these? The genome-wide association studies are very important. These kinds of studies are statistical in nature and benchmark two populations, a population of subjects affected by a certain disease that can be represented with the vaccinal phenomenon and the control group. And so there are a whole set of evidence-based comparisons, the gene expression or the polymorphisms. So to better understand in the detail, we have two populations. One is affected by a certain kind of disease compared to the control, and now the SNP1 is represented in the same way, and so we think that this is not associated to the pathology. Instead, SNP2 is more represented in the population affected by the diseases, and so we think it can have a role in the pathology. Of course, this is a table taken from a review that shows the most important studies in vaccinology, which the first work dates back to 2008. This means that we are really at the beginning from the scientific point of view to approach this kind of field, and the first studies of genetics can be works that lay down the foundations to discover the very first polymorphisms and the role that genetics can play in vaccination. But of course, we are at the very beginning. And so these studies are highly complicated and statistical in nature and difficult to be replicated. So we need to underline also the limitations of these studies and understand that we need time to get to viable data. So what are the limitations? First of all, in these kinds of studies, they have to be confirmed. So we need different replication studies that replicate, because in science it's fundamental to replicate a certain data, and there are statistical difficulties, differences in the control cases, problems with the haplotype mapping, disbalances. So we have many statistical interferences that can complicate these replication studies and the confirmation studies. Moreover, when we have a certain result from these GWAS studies, it's just a statistical association, which is not always translated into a causative or a real confirmation. So I want to say that before understanding the real role, we need to fully understand the impact of the polymorphism from the single nucleotide change, and we need to understand what's its real role. To do that, we need a multi-step process, a path that allows us to understand from the single nucleotide change where this SNP is present, if it is located in a codifying or in a not codifying region, if it's in a protein, what's the role of this protein, and what's the role in a certain biochemical pathway, up to understanding how it is inherited in the population. So, all in all, we have such a funnel situation, so to say. We move from GWAS studies, so it's the funnel is wide on top, and then it narrows down. And so we can start from these GWAS studies that today are fundamental to get the first associations, to get to know the first associations among different genes, polymorphisms, and the impact on a phenomenon that we are investigating. And then little by little, the possible candidate polymorphisms will be restricted because we need confirmations, and so we will perform replication studies, and increasingly we need in vitro and in vivo studies. So what I mean, these data are important, but at the beginning they have to be taken with a pinch of salt. We need to get to confirmation, so it's a long way that separates this research part up to getting to real data, concrete data, to the search for polymorphism that do really have an impact on the phenomenon that we are investigating. So, and then, of course, the possibility to have clinical studies. When we talk about polymorphisms, of course, we do not refer to genetic pathological mutations. The difference is subtle. It's always nucleotide variations, but in one case we have the certainty of having a certain pathology, as it happens with different genetic pathologies. In polymorphisms, there is a probabilistic and statistic association. So, I mean, we can have the probability that can be 4, 5, or 7 times higher to develop a certain pathology compared to the general population. But this, of course, is not certain. So, like in a football match, we have the best player that maybe is going to solve the match, that is going to play a pivotal role, but of course there are other 10 players playing on the field. So it's not always the case that the strongest is going to determine the result. It's a combination of factors, and so this whole study on polymorphisms and the increased knowledge of the genetics can, of course, be of help in understanding vaccinology, so that this could allow us to introduce a breakthrough in the therapeutical setting and to develop new vaccines that might lead to an advantage, allowing us consequently to move to a personalized therapy to produce more efficient vaccines on the one hand and safer on the other hand. So I would like to conclude to reiterate that where are we now at this point? We are at the very beginning of this road, so we need time, takes time before being certain in certain statements. This graph is showing you some bibliographic associations among different disciplines in blue, genetics, in red, vaccinology, in yellow, oncology. What we can see, yellow is blurred because green is prevailing, and green is the union of blue of genetics with yellow of oncology. What does it mean? It means that these two disciplines, these two fields today are strictly interrelated. As a matter of fact, oncology is probably the main field in which we are developing personalized medicine, and we already have today the first examples of therapeutical personalization. If instead we take a glance at what happens between genetics and vaccinology, we can say that at present there is simply these two fingers not even touching themselves. So if they are not even touching, maybe tomorrow we hope that will be this deep hug